This is part 1 of the video focusing on molecular characteristics and mechanism of skeletal muscle contraction. First, I'd like to start off with a little bit of anatomy, getting the first structure down of the muscle, and then we'll dive deep into what actin and myosin kind of do. Starting off, you have a muscle here, this big structure. On the ends of it, you have tendons, and it's surrounded by a layer of fascia to keep it all connected. And going further from that, you're going to have this structure here. It's in a bunch of clumps of endomyosin here, which then specify out into sarcolemma and then an individual myofibril. And within the myofibril, you'll have lines and lines and lines of actin and myosin. A little bit on the purpose of having your thick and thin filaments here. We have this yellow section here, we call that myosin, and these smaller sections here, the little blue and red, and that's actin. Myosin specifically is made up of a couple different materials, but this picture down here is what actin looks like. Actin is made up of a nebulin surrounded by a double helix of your G-actin, which are the little balls here, and on the G-actin there's an active site, but the active site is covered by your tropomyosin. And about every 7 or 8 G-actins there is a troponin. Troponin has 3 different subsections. You have a troponin I, troponin T, and troponin C. Troponin I directly connects to your G-actin. Troponin T directly connects to your tropomyosin, and troponin C is facing outward from the rest of the body. Troponin C has 4 different sites of binding for calcium. 2 of them are strong, 2 of them are less strong. And when those 2 less strong binding sites are activated, it removes or moves the structure of tropomyosin off of the active site of G-actin, allowing for myosin to come in and bind. Myosin itself looks something like this, and there's about 6 of these per section of myosin. So there's going to be 12 heads that connect onto actin that allow the contraction. Myosin has the actin binding sites on the heads and ATP binding sites as well. And then it has 4 light chains, and the heads and the bodies are part of the heavy chains, and they're intertwined in 2s of the heavy chains. And there's a section called the hinge region that cocks and uncocks, allowing contraction of muscles. I think it's really important to note that what is getting shorter is this thing called the H zone, and that's within the sarcomere. The entire sarcomere will get shorter, but the H zone is what's going to disappear as the myosin binds the actin and pulls it closer and closer to the Z discs will get closer and closer, and the I bands as well. And this little diagram here shows if we get a little bit closer. This is a very basic look of what myosin and actin look like in kind of a 3D setting. But these little bodies here are myosin, and coming off of them each is a pair of heads that would then connect to the thin filament, or actin, which grabs on and then pulls on it. And this is actively happening every time you contract your muscles, and it's happening very rapidly. It's not just one contraction of one grabbing of the myosin onto the actin, it's multiple times, and it's kind of crawling its way to close up that zone. I would now like to focus a little bit more on the overall picture, and then we'll dive deep into the mechanism of the actin-myosin contraction. So off of this picture, it's a little bit simplified, but initially there's an action potential that comes down from a motor neuron that releases acetylcholine. Each one of these is a vesicle that attaches to the proteins, these snare proteins, which then releases the contents of the vesicle, which is acetylcholine. And when that is released, it binds to these nicotinic receptors here, which depolarizes the sarcolemma and generates an action potential. When that action potential is increased, there is a release of calcium from the sarcoplasmic reticulum. That happens through these little receptors here that sense the change in voltage from the inside of the cell as sodium comes in and depolarizes, which then opens a gate on the sarcolemma or sarcoplasmic reticulum, which releases calcium into the interior of the cell from the sarcoplasmic reticulum. Once this calcium increases inside the cell, it directly attaches to the troponin C, as we discussed earlier, which causes that conformational change, moves the tropomyosin, and exposes the myosin binding sites. Once that's exposed, the myosin heads are able to now bind to actin, causing a power stroke, and which releases ADP, which we'll go on to a little bit more detail here in a second. This is a little bit more of a simplified picture of what's going on, but here we have the myosin, this line is the actin, and once the site is activated by the calcium, it removes the tropomyosin and allows it to be active. The myosin binds to the actin, and through that binding it releases heat, but also breaks the bond of the phosphate, causing the myosin to do a power stroke. And the power stroke goes from the cocked position to the uncocked position, causing the contraction of the overall structure of the muscle. And then once you add ATP back into the system, it binds to the head of the myosin, causing the break and bond. Well, the binding to the head releases heat, but also releases energy to break the bond between the actin and the myosin. And then through hydrolysis, the ATP then breaks up to a phosphate and an ADP and puts it back into the cocked position. And as long as this active site is exposed and there's an increase in calcium inside the cell, this cycle will happen again and again and again, causing contraction, and will cause the muscle to contract. Returning back to this picture, for relaxation, the calcium needs to leave the interior of the cell, and there's these things called circopumps that are on the surface of your sarcoplasmic reticulum, which will pump the calcium out of the interior of the cell into the sarcoplasmic reticulum. And once the calcium leaves and breaks bonds from the troponin, then it leads to tropomyosin binding back on to the active sites, not allowing the myosin to contract anymore, which then relaxes the muscle. Moving on to part 2, we're going to discuss sources of energy for muscle contraction. So there's 4 different sources, and we're going to start off with 1 and work our way through 4. Starting with 1, it's the quickest and shortest lived way of using ATP. And that's through just floating cytosolic ATP. From here, it's an anaerobic process, and it's immediately available, lasting about 1 to 5 seconds. And it's good for short and quick movements like moving your eyes. Moving on to creatine phosphate. Creatine phosphate is a high energy compound that rapidly transfers its phosphate to a molecule of ADP, which then replenishes the ATP. This requires creatine kinase, and it's mostly located around the M line of the sarcomere, right in the middle there of the sarcomeres. This adds an extra about 10 seconds or so of energy from ATP. Moving on to glycolysis, which is the breakdown of glucose. This is another major source during anaerobic. So the past 3 have been anaerobic activity, and glycolysis gives us about 1 minute. And it's really important because all of these do not require any oxygen. They're anaerobic. But the final and longest lived type of ATP source is aerobic respiration. And that's where your mitochondria in your cells use oxidation of glucose, fatty acids, and amino acids to produce about 30 or 32 ATP per glucose. And this is important because it supports sustained long duration activity. And so things like long distance running or going on long walks, this is where you're going to get your energy from. But this does require oxygen, and it takes a minute to start because you have all these other anaerobic. Alright, moving on to part 3, we're going to talk about the two different types of smooth muscle and the contraction mechanism. What we have showing here is the contraction mechanism of both types of smooth muscle. So the two types you have are phasic smooth muscle and tonic smooth muscle. Phasic smooth muscle contracts in more brief bursts that follow relaxation, and they're found in organs that require more of a periodic movement, like your intestines, your bladder, your esophagus. They often have a display of rhythmic contractions, or something called peristalsis, something that happens in your intestines which helps move food along. And it typically has a pretty low resting tone. And so tonic muscle is a type of smooth muscle that maintains a continuous and sustained contraction over really long periods of time. It is found in things like sphincters and blood vessels and in certain airways. Has a higher resting tone and can maintain tension with a minimal energy use through something we call the latch state. This is regulated by some things that we don't quite understand, but really it has to do a lot with the concentration of calcium within the cell. So starting to go through this mechanism here, we start here at number 1, and you have the calmodulin molecule. It's inactive, but when calcium comes in, it activates the calmodulin molecule, which then allows the activation of this thing called myosin light chain kinase, or we call it MLCK. MLCK is important because it allows your myosin head within smooth muscle to then attach to the actin. And it does this because the MLCK allows binding of ATP to, well, not ATP, but a phosphate specifically, to the light chain of the myosin. And through this binding, it has a conformational change within the head of the myosin to allow it to push the tropomyosin out of the way and allow the binding into the active sites. And so this little phosphate here, this one I'm circling, is the one that's added through the MLCK. And to go on, if we look here at step 4, when the myosin head is phosphorylated by MLCK, it then attaches to the actin, and it releases the ADP and phosphate from before and causes the Cox stage to go into the uncoxed stage, causing the contraction. And then when ATP comes along, it does the same thing as skeletal muscle and unbinds from the actin because of that binding of the ATP. And then if we do have this thing called MLCP come along, which is activated or deactivated by Rho kinase. When Rho kinase is at low concentrations, there's more active MLCP, which removes that added phosphate from MLCK. And that circles back around and causes the head of the myosin to go back into the Cox stage. And if there isn't enough of the MLCP, it is able to contract and relax and contract and relax as much as possible. But if it loses that phosphate, it has to go through that first stage of MLCK to then allow that conformational shift.